In the adult rodent brain, neural progenitor cells migrate from the subventricular zone of the lateral ventricle towards the olfactory bulb in a tract known as the rostral migratory stream, or RMS. The location and characteristics of the normal canine and feline RMS are now reported to facilitate the study of neural progenitor cells and stem cell therapy in large animal models of central nervous system, or CNS, disease. In dogs and cats, diseases such as cerebral infarction, seizures, tumors, demyelination, and neurodegeneration all occur spontaneously, just as they do in humans. 7 scientists from the University of Pennsylvania in Philadelphia conducted a scientific study on how an understanding of the identification, mapping, and measuring of the RMS in the canine and feline brain can further aid in the studies of the human brain in regard to neurologic disorders. The research article entitled Identification of the Rostral Migratory Stream in Canine and Feline Brain was published in the journal PLOS ONE on the 11th of May 2012. The study used 5 colony-raised cats aging from 1 to 3.5 years old and 5 dogs ranging from 1 to 5.5 years old. Each experimental animal, except for cat 1 and dog 5, was administered 1 dose of freshly prepared 5-bromo-2-deoxyuridine, or BRDU, in saline per day for 4 or 5 days and were perfused 24 hours after the final dose. Cat 1 and dog 5 were administered 1 dose of BRDU and perfused 6 hours after the dose. The canine and feline brains were carefully removed, keeping the olfactory bulbs attached when possible and postfixed in formalin. Sagittal sections were then prepared for the study. The RMS was located by the use of immunohistochemistry, nasal staining, and image capture and analysis. To determine if cells in the RMS were proliferating, the thymidine BRDU was administered and detected by immunostaining. The RMS in the canine and feline brains was located and an initial characterization was made from the nasal stained sagittal sections. They appeared as a prominent, dense, continuous cellular tract beginning at the base of the anterior horn of the lateral ventricle, curving around the head of the caudate nucleus and continuing laterally and ventrally to the olfactory peduncle before entering the olfactory tract and bulb. The RMS tract corresponds closely to that of the RMS in the human brain, but is much longer in proportion. The BRDU immunoreactive cells were present throughout this tract. The RMS was also immunoreactive for markers of proliferating cells, progenitor cells, and immature neurons, but not for the NEUN, a marker for mature neurons. Luxol fast blue and CN-PACE staining indicated that myelin is closely opposed to the RMS along much of its length and may provide guidance cues for the migrating cells. In summary, this report identifying, mapping, and measuring the RMS in the dog and cat lays the groundwork for future studies to better understand the involvement of ENPCs in the pathogenesis of neurologic disorders. Thank you. Naturally occurring diseases in cats and dogs have a significant advantage for translational research in that they frequently demonstrate the same clinical signs and pathophysiology as the diseases in humans. The RMS of endogenous neural progenitor cells, or ENPCs, has not yet been identified in canines or felines.